Hello everyone, my name is Saumik Biswas and I'm from the University of Western Ontario in Canada. And the title of today's presentation is The Long Non-Coding RNA HOTAIR is an important angiogenic mediator in diabetic retinopathy. The International Diabetes Federation projects that in the year 2040, nearly 642 million people will be living with diabetes. Now, with an increase in diabetes, there's also an increased risk of developing diabetes-related complications. Diabetic retinopathy is a very prevalent microvascular complication, and it is among the leading causes of blindness in the world. Presently, there's an urgent demand for developing better effective therapies, and in order to do so, we really have to identify and understand new molecular pathways. The field of epigenetics offers those potential solutions and therapeutic targets. Epigenetics is essentially changes in the gene expression without changes in the underlying DNA sequence. Some of the epigenetic mechanisms that are involved include histone modifications, which are histone methylation that is mediated by the polycomb repressive complex 2, which is the histone methyltransferase. As well, there is DNA methylation and the activity of non-coding RNAs. Now, some of you may be familiar with this. This group includes microRNAs and long non-coding RNAs. Now, again, for this poster, the long non-coding RNA of focus is HOTAIR. And we look at what the role of HOTAIR is in angiogenesis during the progression of diabetic retinopathy. So in this poster again, we implement a number of different techniques, which include various cell culturing methods, in vivo techniques, RT-qPCR, FISH, immunoprecipitation, and even a DNA methylation array. So we'll take a look at figure 1. And essentially what we did was we cultured human retinal microvascular endothelial cells in normal glucose and high glucose conditions. And we performed a time-dependent experiment. And we found, interestingly, at the 48-hour mark that HOTAIR, VEGF, and ET1, which is a potent vasoconstrictive peptide, we found that the RNA levels were significantly increased at the 48-hour mark for all three of these markers. So subsequently, for the 48-hour time point, we knocked down HOTAIR using an siRNA, and we found that there were reductions in VEGF, ET1, as well as the components of the PRC2 enzyme. And this is looking at it at the RNA level. For figure 3, we acquired vitreous humor samples from non-diabetic and diabetic patients undergoing vitrectomy. And we found that the diabetic vitreous had increased upregulation, or they had an increased expression of HOTAIR in the vitreous. For figure 4, what we did next is we took normal, healthy human retinal endothelial cells, and we cultured these cells with either the non-diabetic vitreous or the vitreous humors from proliferative diabetic retinopathy patients. And we found, as you can see on the figure on the left, that there is an increased degree of morphogenesis in the cells incubated with the diabetic vitreous. And we show that there are increased expressions of HOTAIR, VEGF, and ET1 at the RNA level. For figure 5, we performed an RNA FISH. So we wanted to determine where the localization of HOTAIR was. And as you can see, that high glucose not only promotes the expression of HOTAIR compared to normal glucose cells, but HOTAIR seems to be localized in the perinuclear region. For figure 6, we performed an RNA immunoprecipitation, and we found that high glucose promotes strong binding associations between HOTAIR and some of these epigenetic enzymes, as I mentioned earlier. So EZH2 is part of the PRC2 histone methyltransferase enzyme, and P300 is a histone acetyltransferase, which is responsible for histone acetylation. Next, for figure 7, we administered this compound known as DesNeb, which globally inhibits histone methylation. And we saw that HOTAIR RNA levels were reduced, significantly reduced, under high glucose stimulation. As well, their VEGF and ET1 were differentially expressed. For figure 8, we performed a DNA methylation array on human retinal endothelial cells incubated in normal glucose or high glucose conditions at the 48-hour mark. And what we found was that high glucose promotes a strong DNA hypomethylation at the hot air promoter region. And what we did next is we administered different DNA methylation inhibitors, which include 5-AZA-DC, as well as an siRNA targeting the DNA methyltransferase 1 enzyme. And what we found was that the hot air, ET1, and VEGF levels were all differentially impacted by these different treatments. So overall, we provide for the first time that a hyperglycemic environment upregulates hot air expressions in human retinal endothelial cells and in the vitreous humors of diabetic patients. So we hope that our findings will encourage further discussion and further research into the role of long non-coding RNAs and their involvement in the progression of diabetic retinopathy. We'd like to thank all of our sponsors and all of our collaborators for funding this great research. Thank you.